Hello dear students. Today we will learn about a feeding mechanism in Branchiostoma. Last class we have learned about the morphology of Branchiostoma, and we know that we have learned how this animal lives or sustains in its habitat, and we have learned the morphological features of Branchiostoma or Amphioxus. So in this class we will learn about the digestive system and the feeding mechanism. Actually in your syllabus it is mentioned feeding mechanism, but in order to understand the feeding mechanism we should know the alimentary canal, its structures, and how actually the food pass through it. So because of that we will learn about the digestive system, which includes alimentary canal and the associated digestive glands, followed by the feeding mechanism. So let us the main objective of this session is to make you understand how this tiny creature like Amphioxus will survive by feeding on this microphagus nutrition, and we will understand the mechanism as to how the food is taken in through the incoming water, how the food is trapped, and how it is pushed or moved from one region of the alimentary canal to the other, and finally how it is digested. So as a prerequisite, you all know the morphology of Branchiostoma or Amphioxus so that you can understand the you can imagine how this Amphioxus look like. We have already seen this photograph which shows how Amphioxus fixes itself in its habitat, putting its oral wood outside and the rest of the body burrows inside. Now coming to the main content, the digestive system, as I said, we will learn under two headings. One is the alimentary canal and the associated digestive glands. Alimentary canal, when we talk about, in case of Amphioxus, it is straight tube and it is complete digestive system. Why? Because it runs from the mouth to the anus, and it is lined throughout by ciliated epithelium, and this cilia plays a very important role in trapping the food particles and moving the food particles from one region of the alimentary canal to the other. Now alimentary canal mainly consists of different regions like the mouth, the buccal cavity, the pharynx, esophagus, intestine, and anus. So let us learn one by one in detail. Now when we talk about mouth, mouth is a large oval median aperture situated anteroventrally below the rostrum or the anterior most tip of the trunk. Now here the key word is anteroventral. Mouth is present towards the anterior tip but towards the ventral region. So you should remember this term. Mouth is a large oval median aperture situated anteroventrally below the rostrum, and this is bordered by a frill like membrane, the oral hood. Now oral hood and buccal cavity. Next structure is the oral hood and buccal cavity. The oral hood is formed by the dorsal and the lateral projections of the anterior end of the trunk. Now this oral hood or buccal cavity contains structures like buccal cirri, or it is also called as oral cirri, vestibule, and wheel organ. Now let's understand what are these oral cirri or the buccal cirri. Now these buccal cirri, they are the the margin of the oral hood is beset with around 10 to 11 pairs of stiff, slender and ciliated oral or the buccal cirri. These are nothing but you can relate this to the tentacles. Like when I say they are slender and they are stiff and they are ciliated. So you have to remember these three keywords. They are 10 to 11 pairs, they are stiff, they are slender and ciliated, and they also bear sensory papillae. Their number increases with age of an animal, and the buccal cirri and the edge of the oral wood are internally supported by stiff gelatinous skeletal rods. It is these gelatinous skeletal rods that impart stiffness to these buccal cirri. Now, what does this buccal cirri do? Now, when the water ingress, like when the incurrent water enters into the buccal cavity of this Amphioxus, it also brings along the food particles through it. So, when the water enters through the buccal cavity, these buccal cirri, they form a sieve or a filter like they all tangle together and form a sieve or a filter like structures to prevent the entry of larger particles along with the food current. Now what happens if larger particles enters through the incoming water? It may choke this Amphioxus. As a result, these buccal cirri and all, they form a sieve-like structures in order to prevent the entry of larger food particles or any sand particles may enter. So it prevents the entry of any foreign particles into the alimentary canal of an Amphioxus. Now next structure is vestibule. The oral wood encloses a large funnel-shaped cavity called buccal cavity or vestibule, into which the mouth opens. So this cavity is lined by ectoderm and it is regarded as stomodium and its external opening the mouth. And next structure that you find is the wheel organ. Now the name sounds, and it may sound unusual to you. Why is it called as the wheel organ? Now what does the wheel do? It rotates, right? A wheel contains certain spikes and then the wheel rotates. In a similar fashion, this wheel organ also has some 6 to 8 pairs of finger-like folds which are formed by ciliated groove, and they are bounded by a ciliated ridge. Now all these collectively form as a wheel organ, or it is also called as rotatory organ or Muller's organ. Now what does this wheel organ or the Muller's organ do? They set in a kind of rotation movement, or they set up a vortex or whirling movement of the water current to sweep the food particles that comes through the incoming water. So here, out of all the 6 to 8 pairs of finger-like folds, the mid-dorsal groove is the largest, which ends in a small depression or a pit on the roof of the buccal cavity. So these are named as Haschek's groove, and the pit that you see on the middle one or the elongated one is called as Haschek's pit. Now these are ciliated, glandular, and they secrete mucus in order to trap the food. Now the next, here you can see the diagram which shows the presence of this oral wood cirri. You can see how they are elongated and finger-like projections along with sensory papillae, followed by the wheel organ, which are finger-like projections. Behind that you can see the velar tentacles, and then which leads to the pharynx, or which leads to the velum, which are in turn surrounded by the velar tentacles, and then from there it goes into the enterostome or the mouth. Now behind this vestibule is closed by a circular ring-like vertical membrane called the velum, which is perforated by a central circular aperture called the enterostome. Now from this enterostome, in some authors consider this as mouth, but then some will say it cannot be correlated with the mouth of the chordates, as it is not lined with the ectoderm. So overall it is an opening from which the water enters into the pharynx. So the posterior border of the velum is produced into 10 to 12 slender ciliated and sensory velar tentacles, and they also serve to strain the water current entering through the enterostome. Now from the enterostome, the water enters into the pharynx. Now pharynx actually occupies the larger area of the alimentary canal. Now if you look at this picture, you will get an idea. Now this region, like beyond the velar tentacle till this atriopore, the whole striations that you see are the gill slits, the pharyngeal gill slits, and this occupies the major portion of the alimentary canal. Now pharynx here is very large, spacious and laterally compressed chamber forming the largest part of the alimentary canal. It occupies nearly 1/2 anterior part of the body, and it remains suspended in the atrial or the peribranchial cavity, which surrounds it on all sides except the dorsal side. Now let's learn about the pharyngeal wall and gill slits. The lateral walls of pharynx are perforated by 150 to 200 pairs of closely and obliquely set narrow and vertical openings called the branchial apertures, or it's also called as gill clefts or gill slits, through which the pharyngeal cavity communicates with the atrial cavity. Now while discussing about the morphology, we learned what is atrial cavity. It is formed by the lateral projections of the body wall that we called as metapleural folds, and that is joined horizontally on the ventral side to form a cavity which is termed as atrial cavity. And then this horizontal lining that joins two metapleural folds, we call it as epipleur. So here the gill slits bear no gills, and their number increases with age of the animal, and it gets added on to the posterior side of the pharynx. Now if you look at a gill bar, each gill bar it consists of a ciliated epithelial covering, both ectodermal and endodermal, enclosing a mesodermal core containing fibrous connective tissue, blood vessels and a supporting gelatinous skeletal rod. The cilia all over these gill bars, they are not uniform. You can look at this picture. Here if you see the cilia all around this gill bar, they are not uniform. They are smaller on the lateral side and they are little elongated on the dorsal side or the apical part of this gill bar. So the cilia that you shorter cilia which is seen on the lateral sides are called as lateral cilia, and the one which is towards the front side and which is little longer, they are called as frontal cilia. Now here in this diagram you can make out that the pharyngeal gill bars, they are of two types, primary and secondary, and both of this they regularly alternate with each other, and they also differ in their structure and mode of development. Now what are the difference? How do you differentiate between a primary gill bar and a secondary gill bar? The skeletal rod of a primary gill bar is called a primary gill rod. Its lower free end is forked and it encloses a blood vessel and a coelomic canal throughout its length. Now what do you mean by forked? It will be split. Now if you observe this picture towards your left hand side, it shows both primary gill bars and secondary gill bars. So if you observe the primary gill rods, it is forked into two, it is split into two, whereas the secondary gill rods, they are not split into two, they are continuous. So whereas a secondary gill bar, it develops as a small tongue bar which grows downwards from the dorsal wall of a primary gill slit. It finally meets the ventral wall so that the original primary slit is divided into two secondary gill slits. Its terminal end is simple, that is it is not unforked, that is it is unforked, means it is not split, and it encloses a blood vessel but no coelomic canal. The primary gill bars are interconnected by a system of transverse bars or synapticula, which impart a basket-like appearance to the pharynx, similar to that of the branchial sac of the tunicates. So this synapticulum also contains a blood vessel and a skeletal rod. Now the next structure that we will learn about is the epipharyngeal groove. Now when we talk about the pharyngeal cavity, three main terms we use, and you should be knowing the correct location of this. One is epipharyngeal. Epi means above, then so the groove that runs towards the upper part of this pharyngeal cavity is the epipharyngeal groove. Peripharyngeal groove. Peri means towards the periphery, and one more is the hypobranchial or the endostyle, which runs along the floor of the pharyngeal cavity. So now let's learn one by one. Now when we talk about this epipharyngeal groove, it is an hyperpharyngeal groove because it is prominent ciliated groove present mid dorsally along the roof of the pharynx. So since it is present on the roof, we call it as epipharyngeal groove. Now posteriorly it runs up to the esophageal opening. Then endostyle. This endostyle is a hypobranchial tract. Why hypo? Because it runs towards the ventral side, or it extends mid-ventrally along the entire floor of the pharynx. It forms a shallow groove with four longitudinal tracts of mucus secreting gland cells alternating with 5 tracts of ciliated cells. The cilia of the median tract are the largest. So if you look at this picture, this shows the TS of an endostyle. You can see that there are gland cells which is alternating with the ciliated tracts. You have cluster of gland cells and the ciliated tract. So if you observe all the ciliated tract, the median one, that is towards the center, is slightly longer. I mean, it's much more longer when compared to the other ciliated tract. Now when we learn about the feeding mechanism, we will know why only this median, or what is the function of this longer median cilia. And according to Barrington, it is this endostyle which is converted into thyroid gland of vertebrates. The next one is the peripharyngeal bands. These are a pair of narrow ciliated tracts arising from the anterior end of the endostyle. They run obliquely upwards and backwards, one along either lateral wall of the pharynx, and they both unite on the dorsal side with the anterior end of the epipharyngeal groove. Now these bands demarcate a small anterodorsal prebranchial region of the pharynx which is devoid of gill slits. Now the next structure in the alimentary canal is the esophagus. Posteriorly, the pharynx leads into the short, narrow and tubular esophagus having ciliated internal lining and leading into the gut or the intestine. Now intestine here is as long as the pharynx. It is suspended from the dorsal body wall by a dorsal mesentery into the atrial cavity. It is covered by a thin layer of smooth muscles and its internal epithelial lining has several ciliated tracts. So cilia plays a very important role in moving the food forward, backward into the other regions of the intestine. The intestine can be differentiated into three parts. One is the anterior, and which is relatively wider part is the midgut. A large blind pouch or the diverticulum arises from the junction of the esophagus and midgut towards the right side, and the lining of the midgut on the right lateral side bears a crescentic lateral ciliary tract, and this cilia beat downwards directing food into the midgut diverticulum. Midgut is followed by a densely ciliated ileocholic ring whose ciliary action rotates the mucus card containing food, and the hindgut that follows, this is followed by a hindgut. If you can look at the picture, you can make out the pharyngeal region leading into the midgut, esophagus, and esophagus leading to the midgut or the first part of the intestine, from which arises the midgut diverticulum and then the hindgut, and there is an ileocholic ring seen at the hindgut region, and the hindgut that follows is narrower and its terminal part or the rectum is heavily ciliated and opens to outside through anus. Now the last part of the alimentary canal is anus, which is a small circular aperture which is controlled by a sphincter. It opens at the base of caudal fin a little on the left side of the midventral line. Now while studying morphology also we said that the anus is shifted towards slightly towards left side, and now maybe you can reason out why the anus moves towards left. It is because of the midgut diverticulum which is extended forward. The anus gets turned towards it gets shifted towards the left from the median line. Now the next thing that we need to learn is the digestive glands. Now here in Amphioxus, the midgut diverticulum, which is often referred to as liver, is the main digestive gland. It arises as a blind pouch from the ventral junction of esophagus and midgut, and it extends forward into the atrial cavity on the right side of the pharynx up to 1/3 of its length, and it is surrounded by a narrow coelomic cavity. Its inner lining has a strong ciliated groove for movement of food. Its zymogen cells secrete a number of digestive enzymes, and it helps in the process of digestion. So this is how the movement of the food takes place in the intestine. This shows the movement of food in the that is behind the pharynx, what happens in the intestine. You can see, you just follow the arrow marks and that will guide you that will show you the movement of the food. It comes from the pharyngeal pharynx to the esophagus, and from the esophagus as it enters into the midgut, the the cilia of the midgut pushes it into the midgut diverticulum. In the midgut diverticulum, the process of digestion takes place, and then the food is moved towards the hindgut. And if you see, the lines are represented as spirally twisted ones because that is how the food get mixed with the help of the cilia. The food is laden with the mucus, which is secreted by the endostyle, and this mucus laden food card gets moved with the help of beating of the cilia, and from the midgut it comes back to the hindgut, and in the hindgut again it gets rotated a couple of times because of the ileocolic ring. Just observe the arrows, and that denotes how the food is moved, and from the ileocolic ring it passes into the rectum and finally through the anus. Now let us concentrate on the feeding mechanism, and in order to understand that, first let's learn what does this branchiostoma feeds on. It mainly feeds on, it is basically a microphagous animal feeding on protozoans, diatoms, algae, and other pelagic microscopic organisms which are suspended in seawater. Now, when you look at the feeding mechanism, like how actually the food is taken in, branchiostoma is a ciliary feeder. It is also a filter feeder. Now, the animal leads a sedentary life buried in sand and with only the anterior body end protruding out into the water. Now, here the food particles are filtered from the current of water that enters through the pharynx, that enters through the mouth into the pharynx, and finally it gets ejected out through the atriopore. Now, the rotatory movements of cilia of the wheel organ, it causes a whirling current of water into the mouth, and the buccal cirri which are present all around the oral wood, it turns inwards to form a fine mesh or a sieve which prevents the entry of larger food and foreign particles. While passing through the enterostome, the water current is further filtered by velar tentacles so that only very fine food particles enters into the pharynx. The chemoreceptors present on this buccal cirri and velar tentacles, they probably serve to test the nature of water current and the food particles. Now, these food particles in the oral hood that escape the main current, which they are caught up and concentrated by the mucus secreted by this Haschig's groove and pit. Haschig's groove and Haschig's pit. Now, these food particles, they are pushed through enterostome into pharynx by the whirling action of wheel organ and join the main current. The ingress of water into pharynx is controlled by the vela. Now, during this process, periodically what happens? The atriopore, which is the opening of the atrial cavity, that closes and the transverse muscles of the atrial floor undergo sudden contraction. Now, when such sudden contraction occurs, what happens? This sets up a reversal of water current. Now, in doing so, the water is pushed back, it's pushed out of the animal so that the water from the atrial cavity is moved into the pharynx, and pharynx it is pushed out through the oral cavity. Now, initially when the water entered, what happened? All the oral cirri and all, they had formed a sieve-like structure. Now, when the water is flowing in the reverse direction, all these finger-like structures, the oral cirri which has formed into a sieve, they all get released. They all come back to their original position, and they are again, and they shed off all the larger food and the foreign particles that are accumulated on them, and again they are set free to receive one more current of incoming water. Now pharynx plays the most important part in food collection. Why? Because it is the cilia of this gill slits which enables the movement of the food particles in the pharyngeal cavity, and in doing so the mucus that is secreted by the endostyle, okay, this mucus which is secreted by the endostyle, they help in trapping of all these food particles in the form of a card. So thereby it results in the food laden mucus that is secreted by the endostyle. It traps all the food particles and forms the food card, and by the continuous beating of the cilia, this food card is pushed backwards. And if you observe this diagram, this will give you an idea as to how the course of the food laden mucus card moves in the pharyngeal cavity. So here what happens, inside the pharynx, the food particles get entrapped in the mucus secreted by the glandular tracts of endostyle and by pharyngeal epithelium. The endostylar cilia and the frontal cilia of the gill bars, they beat upwards so that the mucus sheaths laden with food particles move dorsally to the epipharyngeal groove. The food particles settling down in the peribranchial region of the pharynx are also swept up by the cilia of peripharyngeal bands to the mid-dorsal epipharyngeal groove. So the cilia of the epipharyngeal groove beat backwards, moving the food laden mucus into the esophagus in the form of a narrow food card or food cylinder. Now one such food cylinder leads when it comes to the intestine. Then from the that is the first part of the intestine, that is midgut. From midgut it enters into the midgut diverticulum, where a part of extracellular digestion takes place with the help of enzymes, and then it is moved backwards to the hindgut and then to the rectum. Now let us learn about the process of digestion. Now digestion in case of Amphioxus occurs in both the ways, that is extracellular and intracellular digestion. Intracellular digestion here takes place mainly in the phagocytic cells of the midgut diverticulum, and the extracellular digestion occurs by the enzymes that are secreted by the epithelial cells of the gut and the midgut diverticulum. Now the digested food here, and the main enzymes that are formed or that are found in Amphioxus are the amylase, the lipase and the protease. And this digested food is absorbed partly in the midgut but mostly in hindgut, and the undigested material is finally thrown out of anus. So the main source of these digestive enzymes are the midgut diverticulum and the midgut epithelium. So as the food card from the pharynx passes through the esophagus into the midgut, the lateral tract of cilia, it is directed into midgut diverticulum and driven again to midgut by ciliary action. So the process of digestion starts in the midgut and continues in the remaining part of the intestine. In the ileocholic ring, the food card undergoes rotation due to ciliary activity. This breaks up the food material into still smaller particles, thoroughly mix up with enzymes with food, and delays the backward passage of food to allow more time for the action of the enzymes. So here you should remember the enzymes that are secreted includes amylase, lipase and proteases. So intracellular digestion mainly occurs because of the phagocytic cells that are found in the midgut diverticulum, and also some papillae which are on the floor of the atrium, they also contain the phagocytic cells which can engulf these food particles. So this explains the complete process of digestion and the feeding mechanism in branchiostoma. Now let's learn few questions to know how well we have understood the content. For example, like which of the following is the name given to finger-like projections of oral hood in branchiostoma? So your options here: tentacles, velar tentacles, buccal cirri, or velar tentacles. Now what's the answer? The right answer is option C, the buccal cirri. Now the second question: which one of the following is position of mouth in branchiostoma? Is it anterior, ventral, anteroventral, or anterodorsal? What's the answer? We have stressed about this point during explanation. Now the right answer is answer C, anteroventral. The third question: opening of buccal cavity in branchiostoma is called vestibule, enterosome, oral hood, or the mouth? The right answer is the mouth. One of the essential structure for filter feeding in branchiostoma is called wheel organ, Muller's organ, rotatory organ, all the above. Your answer: all the above, because all means they are all the essential structures which helps in the filter feeding. Wheel organ is also known as rotatory organ or the Muller's organ. Question 5: which of the following statements is true in branchiostoma? Listen to the statements carefully. Mid-dorsal ciliated groove is called epipharyngeal groove in pharynx. Mid-ventral shallow groove of the pharynx is called endostyle. Mid-dorsal shallow groove wall of the pharynx is called endostyle. Mid-ventral ciliated groove is called epipharyngeal groove in pharynx. So guess which statement is true. You should understand these terms by their names. We said epipharyngeal. Epi means the upper one, which lies towards the dorsal edge of the pharynx. Then endostyle, we described it as an hypopharyngeal groove, so you should be knowing the position of it. And then so the last one also reads like mid-ventral ciliated groove is called epipharyngeal groove in pharynx, and the right answer is the mid-ventral shallow groove wall of the pharynx is called endostyle. The next one: which of the following feeding is found in branchiostoma? It should be is, not if, so slightly corrected. Which of the following feeding mechanism or feeding type is found in branchiostoma? Microphagy, coprophagy, macrophagy, or polyphagy? What's the answer? We said they are microphagous, feeding on minute organisms, so they are microphagous in nature. So thank you for listening to this lecture. I hope you have understood the concept of digestion and the feeding mechanism in Amphioxus. So as an outcome of this session, you should have learned the plan of digestive system of this branchiostoma, and also you will have a idea as to how the process of filter feeding occurs, and these are the references from which the diagrams and the other contents are taken. Thank you for listening.